Good morning. I'm Ian Levine, co-founder and CEO of ModernVivo, where our mission is to redesign the preclinical drug development process. Because the drug development process is costly and ineffective. It costs between $1 and $6 billion to take a single drug from the lab all the way through human clinical trials, where they will fail over 90% of the time. And almost every one of those drugs that failed in the clinic showed success in a preclinical animal study. And this begs the question, why do so many of these trials fail? Well, an abundance of literature suggests the reason for these failures is that these preclinical studies have major design flaws that lead to unreliable data. And to show you why these design flaws occur, I want to take you on a scientist's journey to discovery, because it's a slow and tedious process. A scientist will first test their novel compound in cellular-based assays, and when they see some form of success, they will move on to in vivo animal studies. And when they see success there, they will end on a human clinical trial. But that process of designing an in vivo study is a slow and tedious process. First, they will start by searching databases or for the model of their interest, where they will find thousands of papers that have published their model in the past, where they will go and read about 10 to 30 of those papers and extract important conditions from that pieces of literature, where they will go on and devise their experimental plan in about 2 to 4 weeks. And this process has remained unchanged for decades. But don't take my word for it. Listen to other scientists in the drug development process talking about this slow and tedious process. They know it's slow, that it takes between 2 to 4 weeks to design their experiment. It's a challenging process where they're asked to design experiments often that they're not knowledgeable about. It's an archaic process based off convenient design decisions, not ones optimized for clinical success. And it's high stakes because billion-dollar decisions are made off the back of this information. And this is a pervasive problem across a large market for our 3 core customer groups: pharmaceutical companies, contract research organizations, and academic labs and drug discovery cores. We estimate that over 10 million in vivo experiments are developed every single year, and the spend on those studies will be over $24 billion by 2027. So what we're developing at ModernVivo is an AI-powered protocol design software. As I alluded to, the status quo is slow and tedious, where a scientist will read about 1% of all papers for their model, where they'll design their experiments in about 2 to 4 weeks. At ModernVivo, a researcher simply queries our technology, and they are given an optimized set of experimental conditions where they can design their experiment in as little as 1 day, where we can reduce protocol development time by up to 96%. And to get a little more specific, we use AI large language models to go through every single paper that has ever been published using that model of interest, where we will extract crucial information, and then using internal statistical tools to look at the results of human clinical trials to identify, is there a preclinical model set that is more translationally successful than another? And to date, for technological development, we can extract protocol conditions over 100 times faster than manual review, on par, if not better, in accuracy than manual human review. And to show you what this looks like, a scientist will simply type in the name of the model that they want to optimize and design, and use our optional advanced search tools. And what they are given is every single way that experimental model has ever been conducted, with all the information that they need to design that experiment. Something as big as the genetic strain of the animal, all the way down to the light-dark conditions in the feeding schedule for these behaviors. They're also given the optimized set of conditions. They can identify study sponsors and filter by study design for what they already have on hand. And what we're really excited about is scientists are committing. We have 3 signed LOIs from 2 academic labs and 1 pharmaceutical company, and we have 6 more interested pilot partners that we've been in discussions with. And what's really exciting is we've spent no money on marketing to get these initial LOIs. And don't take my word for it, listen to what scientists are saying, that we need to build this product right now, that our product would be a potential needle mover for their technology and their companies. And companies derive value from ModernVivo in 2 key ways. First, by immediate savings of over $1 million per drug project, by first reducing the number of animals that are necessary for drug development campaigns, reducing the overhead costs of maintaining those animals, as well as reducing scientific consulting costs. And because we're accelerating the drug development process by about 2 to 4 weeks, we can actually extend the patent life of drugs that will end up being approved and lead to over $11 million in increased revenue. So said in another way, ModernVivo can provide over $1.3 million in value annually for every drug in a pharmaceutical company's pipeline. And our business model is tailored software licenses on a per animal model basis. If you're a small pharmaceutical company and you're devising a very limited number of studies, we charge $5,000 for every model developed using our technology. And that price goes down as you scale up. If you're optimizing hundreds of models, that price goes down to $2,000 a model. Now, CROs are a little different because they're client-facing. We charge them a flat rate of $250,000 a year for an unlimited number of models developed. And academic researchers, we charge $2,500 a year for an unlimited number of models developed for their entire lab. And our go-to-market strategy is a strategy with a proven track record. We've talked with other companies in our space and found that what I'm about to tell you is the most effective way of getting our technology into the hands of customers. Our first phase will be targeting academic researchers and small pharmaceutical companies. We'll then move on to medium-sized pharmaceutical companies and contract research organizations. And then within 1 to 2 years, be targeting large pharmaceutical companies. And we'll be doing this in 3 key ways. First, as I alluded to, by having that low price for academic researchers. Once they leave academia and enter industry, they know the value of ModernVivo and bring our technology with them at a higher price point when they enter industry. Second, we want to partner with biotech accelerators and university tech transfer offices to offer a discounted rate to their portfolio companies to have access to a large number of customers very quickly. And then last, we will be hosting outbound product demos and lunch and learn sessions to have a large number of customers in one room at one low-cost event. And what we're really excited about is our competitors operate in different stages of drug discovery. There are companies such as Valo and Benchside that are optimizing in vitro cellular-based research, the stage before ours, and companies such as Trials AI and Oken that are optimizing human clinical trial design, the stage after us. But to the best of our knowledge, we are the first and only company using AI to optimize the design of in vivo studies. We've already filed our provisional patent application and are working on filing our non-provisional at the moment. And the question we get all the time is, well, what about ChatGPT? Why can't it do it? Well, we tried, and it writes terrible protocols. But second, it provides no rationale for scientists. When scientists are writing applications to the FDA or the NIH for a grant, they need to say why they've chosen that particular animal model and animal study design. We give scientists that information that ChatGPT just doesn't do. And our advantage is for our expertise in vivo study design. I've personally spent thousands of hours studying design methods from a variety of in vivo models and have personally read over 5,500 papers and extracted crucial design details from these studies. And this work enables tech development, where we build software to the specific needs of in vivo scientists and can uncover crucial aspects of model design few others understand. And this creates a high barrier of entry for potential competitors. To date, we've raised a little under $150,000 from a variety of sources and are currently raising a $1 million seed round, and this will accomplish a few key details. First, we'll file our non-provisional patent application, be able to hire our full team on track for a Q1 2025 launch, where we aim to have 30 customers and $100,000 in growing revenue. In terms of financials, we'll see early and sustained profitability. By Q2 of 2027, we'll be profitable with a very limited number of clients, and by end of year 2030, we'll have over $68 million in revenue. And to talk about our team, as I said, I'm Ian Levine. I'm the co-founder and CEO. I have my bachelor's degree in experimental neuroscience from Virginia Tech, and I have a master's degree in pharmacology from the University of Michigan. My co-founder Colin is also our CTO of the company, and he has his bachelor's degree in computer science from the University of New Hampshire and a master's degree in biotechnology from Brown University. We have 3 members of our advisory board. Dr. Ann Gregus and Dr. Matt Basinski are our scientific advisors and are both assistant professors of neuroscience at Virginia Tech. And Dr. Wendy Weber-Nelson is our business strategic advisor. She's currently the chief business officer of Danforth Advisors, a life science consulting group in Boston, and has over 25 years of operational experience in the biotech space. Thank you so much.